Morning everyone, so I'm Maxime Reverchon. I work from SYSAAF in France, and I'm working in the field of biotechnology of reproduction in poultry. And today I will present you an overview of the poultry industry, the genetic selection, how it's done, and the different selection criteria that are adopted. So first, my company is called SYSAAF, so it's for French Poultry and Aquaculture Breeding Association. Its specific status is a trade association. Our main objective is to give technical support for the members of our association, that are 37, in the field of reproductive biotechnology, and managing and/or genetic improvement for the different species that are from poultry, aquaculture, and also, since now 3 years, we started with entomoculture species. Another objective is to carry out the label rouge audits, and we preserve the quality and diversity of the national gastronomy with the different products like oyster, truffes, caviar, foie gras. So now I will present you the French avian industry organization and the main actors in this avian industry. So it's a pyramidal organization. At the top there is the pure breed that are called the animal in pedigree selection. It's at this stage that we generate the genetic progress. Then at the second level, there is grandparents and parents flocks where we are spreading the genetics. It means that we are multiplicating a lot the animals in order to spread the genetic progress for the farmer. And at this level, it's the level we sell the one-day-old chicks to promote the genetic progress, or for the layer, or for the broiler. So at the top of the pyramidal organization, there is really few companies. I will present it to you just later. They are the breeding companies. Then there is a multiplier, and at the down of the pyramidal organization, there is a farmer, and there is a lot of companies. So the main breeding company are international leaders. There is the EW group. It's the biggest one. They are doing laying egg breeding stock, vaccine for poultry. They have other activities. And what interests us now today is they have the avian groups. They are doing chicken breeding stock, turkey, broilers, and more recently they bought Hubbard. It's a broilers breeding company. And last years they bought Novogen. It's a layers breeding company. So you can see this group control a lot of genetic animal production. They are in direct concurrency with Hendrix Genetics. Sorry, I couldn't find a better image. But Hendrix Genetics, it's the same. They are really big. They are producing feed, aquaculture, pig, turkey, and also layers and broilers. Then there is another international leader that is called Grimaud. This one is started with two farmers that started to breed Barbary ducks, and then they increase a lot, as you can see. And nowadays it's big groups. They produce a vaccine. They do also genetic selection in waterfowl, pigeon, Guinea fawn, rabbits, pigs, shrimps. They also do vaccine. And since few years, they started to do new protein resources like insect. Oh. What? Oh, okay. It's me or it's you? Oh. It's me? All right, great. And so, between these companies, there is some of them that are members of SYSAAF, my company. I will present it to you now. So For the broilers, we have Ubar from the group Aviagen and, oh, it's me? Okay, I continue. And Sasso from the group Hendrix Genetics. For the layers, there is Novogen from EW Group and ISA from Hendrix Genetics. Then we have also the species duck, geese, pigeon, and guinea fawn from the group Grimaud. Also, we have for duck and geese the group Orbia. We do also some pheasant and partridge genetics with the group Gibaud Vendée. There is 2 companies that are doing quail, but nowadays more Kai Robin is working. And there is another center. This one is really specific because Selection Vechan Center is producing the breast genotype. I don't know if you know it. It's a really famous one, and it's the most expensive chick in the world, actually. And another objective of this company is to maintain some local breeds. So they work with some small association to help them to maintain the genetic diversity in the local breed in France. Okay, so now I will present you the feature of the pyramid scheme. So there is a very wide distribution. At the top, if we take one rooster from the pure breed, you will produce with this rooster 50 chicks in the grandparents' flocks, that they will produce 3,250 animals, that then at the commercial level will produce 275,000 hens, and that these hens they are able to produce 85 million till 100 million eggs. So you see it's a really wide distribution. The problem is that it's really long to disseminate this genetic progress because it takes between 3 to 4, sometimes 5 years from the level of pedigree selection till the commercial chicks for the farmer. So how the pyramid organization and crossing is done? The breeders, they improve their population. Here at the top, known as pure breed. And how they do? For example, here they have 4 different breeds: the A, the B, the C, and the D. And they will cross them together, A and B, C and D, to have the grandparent flocks. Okay. Then they will cross the grandparents together. It's called the multiplication. At this level, they will produce a lot of animals. And with this cross, so you have in one cross the 2 first pure breed and the second one the 2 others. You cross them together again to obtain the terminal product, which is a mix between the 4 pure breed, and in this terminal product that is sold to the farmers. It's a one-day-old chick that they will rear and that will produce or the broiler for the meat or the layers for the eggs. And it's like this if you want that the breeding company, they protect their genetic, how they make their money, because if you want to reproduce this terminal product together, it's almost impossible to go back to the first pure breed, or it will take years and years. And in this pyramidal organization, the problem is that the only measurement available are the ones that are carried at the pedigree selection level, because there is not that much feedback with the farmers to the breeding selection. So to resume, the pyramid organizations enable genetic progress to be disseminated really widely. There is a really hard and intense competition between breeding companies. They're really paranoiac. You can't speak that much about what they do to each other. It takes 3 to 4 years for genetic progress to be spread to the farmers. So it means that if nowadays we speak about we want to improve this character, we want to improve this behavior, we need to anticipate it a lot because before it arrives to the farmer, it will take a long time. And the breeders work on pure breeds that are not accessible to the farmer. Now I will present you the evaluation of the method for genetics. Before 1960, all the genetic evaluation, they were done, it was a massal selection. Whoa. It was massal selection. It was based on the phenotype mainly. Then from 60 to the 90, the pedigree selection started, so they take into account the relationship, the parental assignment between the individual, plus some genetic correlation. Then from 1990 to 2000, they include some trial, meaning they test the animals they produced and their performance to do the selection scheme. Then in between 2000 and 2010, they include the BLUP. To summarize, it's a method of calcul with computers that calculate a lot of statistical equation, and like this you can incorporate a lot of data. So it increases a lot the genetic selection. And since 2010, they are doing genomic selection. So genomic selection, they use marker on the DNA of all the genotype. And this technique is really deeper and allow to improve the genetic value of the animal faster. So today, there is no more breeding companies that use the massal selection. They all work with pedigree, and majority of them nowadays, they include the genomics. So now I will present you how is done the breeding in a breeding center. So the breeding company, they select the animal according to the objective of production. There is different selection criteria, growth rate, meat yield, yielding rate, equality. Nowadays there is new challenge like food efficiency, meat quality, laying persistence, disease resistance, laying in nest also, and I will come back to it later. So the pure breeds, they are reared in the specific center. It's like this, it's building next to each other. To enter, it's really complicated, it's really restricted. You need authorization, you need to didn't be in contact with birds 3 days before you go in the center. At the entrance you need to take a shower, full shower. They give you clothes, even underwear to enter in it. And in each building there is a different breed, like the ABC I show you from the start. Then the adults, they are kept in an individual cage, and they record the performance, the phenotype of each animal to estimate the genetic value of each individual. So a pen is made with one male that will be coupling with some selected female according to their performance. So like this, we control the reproduction to know which male go with on which female, and to produce at the next generation family of full brothers and full sisters. So how you do that? You will collect the semen individually of each rooster. Then you will do artificial insemination according to the selection scheme. To be sure that you don't do mistake, they have adapted the cage. Like for the layer, for the hen, they have receivers that are separated from each cage to be sure the eggs is from this hen and not from another, to don't mix it. And plus, when they collect the eggs, they write the number or of the cage or of the animals, because they all have wind bank. Then they store it. It depends. For the broiler, it's maximum 10 days. For the layers, it can be longer. and they place it in an incubator to start the incubation to produce the embryo. After 2 weeks, after 2 weeks, they will mirror the eggs to check the fertilized eggs. Here you can see the blood vessel, so it means there is an embryo from the unfertilized eggs, and they will transfer it into the hatchery. So here, at the transfer, all the eggs from the same hen, they are grouped in the same basket to finish their incubation, and on the top of the basket, they put a top to block the chicks to be sure they don't mix at the hatch. So the hatching is organized into several stations. There is some people, they open the box, they take the birds, they put the wind bang, they do the vaccine, and another one is enter all this data in a big database. So yes, the chicks are, for the correspondence, they are in the same basket, as you can see here. They put a wind bang direct at the wing, really often. And then the chicks, they are placed in a specific building to rear them. And we start to do the first phenotype from the weight, or it can be the behavior, really soon to have the first genetic evaluation to know from each animal their value, their genetic value. And it's the first selection, it's a young choice, as we call it, like this, who already separates the ones that will participate to the next generation from the others. So for example, you can weight the animal, you can sex them, there is also some mortality. So as you have family of full brother, full sister, you can know if the parents, they give a robust animal, and if they can survive, if they have good weight, and if they eat good, and they behave good, all right? Then when the young animals, they are selected, you read the wing band and you place them into an individual cage as before. And then after, you rear them in it, you record their individual performance, and you start again. So the animal selection involves that you have to identify the male and female breeding stock with the highest estimated genetic values. It's what you want to give progress for the next generation, to improve the growth and production performance of the animals that will produce animal protein. How you carry it out? The selection is based on observation of animal performance. This selection requires individual recording on the different parameters you want to phenotype. Once this data has been collected, the result of these performance parameters, they are analyzed. It's a big statistical analysis, big calculation done by computer nowadays. And the animals which present the best results, they are chosen to breed the next generation. Like this, it carries the improvement for the next generation. And with this work, we can create selection scheme that we propose to the breeding company, and they done it to spread the genetic progression. So to do that, they need to phenotype different selection criteria. The adult measurement is carried out according to the selection objectives, which is different according to the breeding company. For the layers, for example, of course, there is the egg quality. Here I present you the main one I'm allowed to present you, but you have to know, in average, there is between 17, 20 different parameters they look for egg quality. So there is the egg size, the shell color, cell strength, the hot unit, it's the height of the albumin, the meat and blood spot, dry matter. They also select the animal for their productivity and persistency. So the goal is to have a persistency improvement. The dream would be to follow the red line, to have layers that produce more than 90% eggs until 80 or 100 weeks production. They work on breeding and food efficiency. They do selection on egg number, peak and persistency improvement. There is a selection on bird ability to adapt to their feed intake and to the different conditions, different feed, different environment. And the goal is to increase the egg number while preserving the fin capacity. They are also selected on their social and calm behavior. So the animals, they are in collective cage or sometimes on the floor, depend. There is a full big bird challenge. So for that, they manage different zootechnic parameters like the light, temperature, feed presentation. They measure the picking behavior and they select the calm families as there is cages or pen with full brother and full sisters of the same family, and they can select like this. They look also the laying behavior. So they put the pen selection in real condition of production. They have individual information through the RFID technology. And now the new breeding criteria they are looking for are laying rate in the nest, the time the hens spend in the nest, time inside they go from one nest to another, if the time between two oviposition increase or decrease, and also the bird's behavior. And all that parameters, of course, they put it in relation with the shell strength, egg size, and shape, the first I show you at the start. For the broiler, they look for the meat yield. So for the white, for the leg, I didn't detail it now. They look also the meat quality. They measure the color, pH, tenderness. And so to do all that, as you saw, we need to have individual performance and to put animal in individual cage. And that is not really ethically good. And citizens, they push to have animal on the floor and less individual cage. With the new legislation that forbids the cage, we need to adapt how to do the selection. And one tool to do that is the genomic selection. So now I will try to present you the genomic selection. So first, I don't know if you know well genetic or DNA how it's done, so I will do few reminders. So genetic is transmitted by the parents through the DNA, and the DNA is compacted into chromosome. I make an image for you. It's like you can consider the DNA as a long TV show, and each episode is a chromosome, okay? And these chromosomes, they come in pairs, and the total number of chromosomes, they vary from one species to another. For example, for human, there is 23, but for chicken, it's 78. So, and one chromosome, so this is a chromosome, they come by pair, so, and each chromosome is composed with two alleles, two strands if you prefer. So the DNA is a secure instruction, and it's written in universal languages. This means that the two strands, actually, they carry the same information that are face-to-face. It's like a security safeguard. The A always faces the T, the C always faces the G. This letter, A, T, C, G, they are called nucleotide. I hope I don't lose you with that explanation now. So all these letters, they are found in the DNA of all living things, plant, animals. And these letters, they have the same meaning in all living things. We speak about the universal genetic codes. So how the DNA passes from one generation to another, it's within each pair of chromosomes. One will be inherited from the father, and another from the other from the mother. The problem is that at the time of reproduction, you don't know which chromosome pass from the father and from the mother, and which allele, which half of the genetic from the father you get, and which half of the genetic of the mother you get. You will see here. Here, there is, for example, for the father, if we take only 4 chromosomes and 4 for the mother, and we don't know how they will be disseminated in the next generation, you can have 16 different possibilities. And here it's simple. I don't talk about other processes that can mix a part of this chromosome with this chromosome that increase the possibilities. So it explains the diversity that can be observed within a family. Okay, it's like this we can do genetic selections because you don't get the same genetic heritage from your parents. So the genomics provide access to all the entire genetic code. On this DNA, the different little letters I show you, there is something called SNP, for single nucleotide polymorphism. It's a marker. It's a marker of single polymorphism of this kind of this 4 letters. A SNP, it occurs when you have a long series of the same letter for each individual, but you have only one that is variable. It's really like a bookmark. So generally we work with biallelic SNPs, for which there is only 2 possibilities. All the time the A with the T, the C with the G, okay? So the SNPs, they are used as a DNA marker. It's a bookmark of our instruction, and the SNP, how we do to read it, we use something that is called a chip. The chip, I did it in French, sorry. The chips will allow us to read the information of the marker and to see if there is difference or no. Like this, I will be able to know if you are full brother, half sister, cousin first, second, third degree. So the chip is a reading tool that will search the SNPs that are coming from a pre-established list. We say, okay, we know that this one, for this species, they are from this family and for the other family. And it will look for each individual and each SNP to see the different version we have. It's like this we can do the parental assignation. So how it works? The flanking sequence of the polymorphism, so the different letters, they will hybridize to the chip, and after there is just a fluorescence measure that is done. And like this, the chips allow you to do the parental assignation. The chips can contain 100 SNPs or 1,000 of 100 SNPs. More you have SNPs, you have to understand that easier it is to calculate the parental assignation. But there is a cost. You can lose €50, for example, if you decrease the number of SNPs. And the chips are species-specific, meaning there is one for Gallus, one for ducks, Barbary Pekin, one for goose, one for partridge. So the genomic selection, the advantage is it's time-saving in the selection scheme because it shortens the cycle. Why? Because when you get your chick, you can take a blood sample after 10 days, 2 weeks, and already calculate the genetic value and know if you need to keep it and rear it, or if you can eliminate it because it will not participate to breed the next generation. It allows the animal to be reared on the ground because you don't need to put them in single cage, as at the end you can do parental vaccination just with a blood sample. So nowadays, what they do, the breeding companies, is they rear the animal in groups on the floor, male and female together. Generally it's 1 male for 5 female. They can have like this natural reproduction, female can lay in nest, and with the blood sample you can so classify the individual within sibling. And I didn't say that, but what is super interesting is, for example, if you want to select roosters on this capacity to produce good layers. Before, how you do with pedigree selection? You make this rooster producing the layers, and you let them lay. So you have to rear them until sexual maturity, let them lay, and record the performances for each layers. And then you say, okay, this rooster is good to produce good layers. Nowadays, with the genomic selection, as we have the performance of the parents, the full brother, full sister, the cousin, we already know before to do the reproduction that he will produce good layers or not. So you win a lot of time. And also, we are able to look for the best individual in different families. So like this, you can keep a high genetic diversity in your pure breed population, because the challenge is to avoid the inbreeding in the pure breed population. But there is also a limit for selection. You can't improve all the time and definitely the performance. There is physiological limits, and for the broilers, there is, for example, meat default that appears, like white striping, wooden breast, or also spaghetti meat. And to finish, I will present you the most commercialized genotype in layers and broilers in Europe. So mainly for the layers, there is a free genotype. There is layers that produce brown eggs. There are the Rhode Islands. There is layers that produce the white eggs. There are the Leghorn. And there is, in really a few percentage, the local breed that are really specific. And for example, there is some of you, they know the Marans. It's a local breed that produce eggs with the eggshell, as you can see here, that look like, that is chocolate color. Here is the repartition in Europe of the eggs consumption. So you can see there is a majority of people that prefer the brown eggs, especially in east of Europe, whereas in the north of Europe, for the Denmark, for example, they prefer the white eggs. And it's really in a position that we can see in the US. In the US, they prefer eating the white eggs. What you have to know, it's a brown eggs or a white eggs. It's exactly the same content of protein, lipid, everything in it, all right? It's just the egg color that change, and it makes a lot of benefits for the market, but actually it doesn't change nothing for the feed consumption for human. Then for the broilers, there is two main categories. There is rapid growth, that are called also conventional, and the slow grower. So the rapid growth, they represent the really majority of the volume that is sold. There is 97% in the worldwide and 91% in Europe. And the production is based essentially for first the economy. It's really economical to rear these animals for their growth, food efficiency, and meat yield. The second category, so they are the slow grower. So it's a really small volume in many countries. These products they are characterized by a specific phenotype and a particular set of specification. There is like the local breed, the organic, the label, farmhouse, certificate chicken. This means they answer more to citizen expectation, and it's more complicated, but there is more regulation to rear them. The main problem for this breed is like the production price is really higher than a conventional one. It can be 15% to 200% more expensive, so that's make a big difference. And after that's why the consumer don't buy a lot this product. And another problem is environmental impact, because when you grow, for example, a conventional broiler like a Ross that grows in 30-40 days, that reaches 2 kilo, 2.5 kilo, compared to slow growing that we grow during 80, 90 days, sometimes more for the local breeds. It will consume more food, so it needs more resources. It will produce more pollution with the dejection. And at the end, economically for the farmer, it's complicated because they have to sell it higher price, and the consumer is not always okay to spend more money for it. I think I'm done. Thank you for your attention.
Thanks. I have a quick question. So you mentioned that there's like 3 main breeds at the end, but I'm wondering from what you showed, and maybe I'm very naive on the topic, but either through pedigree selection or genomic selection, it seems that the whole starting block is always the same, where chicks are produced or selected within indoor environments. And I was wondering how representative that is for outdoor or free-range breeding, and whether there's any kind of, if you know of any kind of breeding selection programs that actually combine, for instance, genomics and free-range conditions.
Yeah. You can use this one.
This one?
It's
okay,
not comfortable.
Pay attention.
Okay. Yeah, actually there is no breeding companies that work with free-range animals. The main reason is before they were keeping all the animals in cage in closed building. Nowadays there is also the avian flu that can be terrible. Even in the pure breed, locked in the building, and as I was saying, you take shower, you don't see birds before going. There is 2 years ago some breeding companies that get contaminated by the avian flu, and it's a pure breed that they have to eliminate all the population. It happened in ducks, and that's why nowadays, I don't know if it is in the other country, but at least in France, there was the product from ducks, so the direct the meat or the foie gras. The price increased a lot because they were contaminated by that. And after to spread it, as I show it to you, it will take 2 years, 3 years. So for now, the price is really high. Then after, it's true, if you want to rear your animals, even your dual purpose or local breeds, in organic or outdoor system, and they are selected in closed building, how you can know that the behavior inside a closed building will be the same outdoor? That's a real question. Maybe there is some local breeders in organics that do their own selection, but I don't know if they have the resources enough to manage it like this, because in my company there are 6 geneticists to work on it, and it's a long time of calculation, statistical analysis to produce the scheme selection. So I don't know, but what would be good is sure that we could think about it. And I don't know, someone yesterday told me that there is a new legislation now. Maybe there will be an obligation for the organic production to have the breeding also in organic systems. Yeah. So that's a point they will discuss, but the one I am working with, for the moment, they don't do that.
I was just curious about, you said that at the breeding level they could identify the behavior of the animals using IPD tags, so they could identify when they were in the laying, where they lay the eggs and where they were. Could you explain a bit more how they do that?
Yeah, sure. It's when they are in a single cage, no, of course.
No.
But now with the genomics, they put the animal on the floor, and they adapt the system to put nest at 30 centimeter, 1 meter. The organization, how they are disseminated into the pen, and the animals, they all have chips on them, and like this you can see when they go inside the nest, how much time they stay. Now there is artificial intelligence with a camera like this. You can see if the hen lay an eggs or not. You know exactly the time she went the day before till the next day, so you know the interval between the two laying time, and they calculate that. You can see also if they go in the same nest or if they change nest. If there is one nest where there is more hens that goes, because it is sometimes more hide, you know, they put some caoutchouc or plastic stuff in front of the nest because they like to be quiet when they lay. And it's all these parameters they measure nowadays, they're on the floor. And there is a PhD student, it's Fleury, maybe if you're interested, he published a paper on it. And he calculates the heritability of this behavior, and there is. So you can select the animal, the family of animals that are more adapted to go to lay in nests or not, because also there is some others that don't lay in nests, they lay on the floor and it's a loss for the producer. And he has really good heritability around 0.2, 0.3. So in few generations, yes, we can have some kind of breeds that are more adapted to be reared on the floor than in your cage.
So they can actually make a heritability on, for example, the ability to lay the egg in the nest and not on the floor. So that can be a trait in the future.
Yeah, totally. They do it. They already do it. They started it like the genomics. I said 2010 the technology, but before they put it in place, it started really like 2018, 2019. So it's already 3-4 years they are doing that. And now, yes, they have some breeds they are selected for, and now it's more than 90% they lay in the nest. So that's really good performance, yeah.
So anyway, we can conclude that free-range animals, so the stocking breeding of free-range animals are not selected starting for the free-range activity, so for the free-range adaptation. So it's
a problem.
Yeah, a pity. I know for sanitary reasons, there are a lot of reasons anyway, but it's a big, we have to probably
improve that, yeah, because after if you rear the animal in another system, are they well adapted or not? Can they do develop their maximum genetic performance in your system, whereas they are not used to that?
Yeah.
You don't know. Thank you.